and look at how we can design a good primer. So this is how the software in the PCR, I mean real-time PCR looks like. In the first tab, you have the setup. In the setup, what you need to define here is that what you are going to do. So in this particular example, we were actually focused on doing the standard curve, which is for the primer efficiency. So standard curve is selected. In the define, what happens is that what is your gene name? We are using cyber green, so it is labeled as cyber green. And then color code you can also change, otherwise a machine will take a color which is reflected in the amplification curve, which I will be showing just now, right? Another thing is that now this is the plate. This is how it looks like. It is having 384 wells. Now in this 384 plates, well plates, you need to specifically know that where you have loaded your sample. So in this example, which is already characterized and you can open and check. In the C1, it is having sample 1. And sample 1, if you click on this, it will tell you it's sample 1. So sample name is 1 and it is having GPX gene, right? And it's in triplicate, right? Second is that, so in the primer standard curve, you have to use 10-fold dilution changes, right? So 10-fold diluted samples in the second C5. You will see that in the C1 you have 1, it means 1 microliter, let us suppose in concentration, or let us suppose 1000 copy number of 1 gene. So in the second, it means in the C5 lane, well, you have again the GPX gene of sample 1, okay, in 100 copy number. So 1000, 100, 1000 copy number, 100 copy number. And similarly, you can have 10 number, 1 number, and then it is very important to keep a negative control, right? And so this is how you label the well. Do not worry that how the well is labeled, because I am pretty much sure if you are working on the real-time machine, definitely you may have a software of similar kind of layout, right? And then finally you have the run method. In the run method, you can see that what is the PCR profiling. So how much is the initial denaturation, then the number of cycles. So 40 cycle was done for this particular PCR. What is the initial denaturation, amplification, elongation, and followed by a melt curve, which helps us to understand the primer specificity, right? Now, after you've done the setup, you will see that in the run, you have several plots, right? You have the melt curve, you have the temperature plot, and so and so on. So let us not go into the run. We will directly go into the analyze. You select in the corner of this, I urge the participants to not worry about the recording. If needed, we will again retelecast the whole episode tomorrow or on a time which we will be sharing with all of you. So kindly focus at the moment on how we do the analysis, right? So you select in the corner of the plate. So in this place where the red cursor, you might be seeing where I am clicking, right? And then you do the analyze. What it does is that it gives you amplification plot, right? You can select for all, and then you will see a lot of noises, background noises, and then you can see the amplifications. So this is how your amplification curve looks like. Let me okay, so this is how the linear graph looks like, where you have, if this Quant Studio is not working, maybe there is some issue with the specification of the laptop or the PC. So kindly focus right now what I am showing. Most of the analysis we will be doing in Excel sheet. So in this linear amplification plot, you will see that up to 24 or 25 cycle, it has not crossed the threshold. So it is very difficult in the linear graph to find the CT value. So to do that, to check that, we convert into the log value. Now you see the same graph is converted in, it's in a much better visible way to look at where the CT value is. So here somewhere around 25 there is a CT value, right? So similarly it's there for all the genes and for all the dilutions, 1000, 100, 10, 1 and then negative control, right? So this is how it looks the amplification plot. Now in the standard curve, this particular example, the reason behind to give this particular example, that it is having all the different varieties of amplification you can think of. For example, for GPX, you will see that the slope value, this slope value is minus 3.5, right? Whereas R square is 0.99, efficiency is close to 91. So which can be used for the gene expression studies. In the second one, for tubulin 1, again it's around 91% efficiency. Tubulin 2, around 103%. And then VVOP. So it is close to 165% efficient, which is practically not possible. For any machine, the maximum efficiency could be 100%. It cannot go 164%. So in this case, you may have to. Hello, can you hear me? Okay, fine. So in this particular case, what happens is that it's 165% efficiency. So it indicates that this particular primer is not good enough to be used for understanding the relative gene quantification of this particular gene. So you need to redesign the primers. Okay. Now let us go. So this is the standard curve from the software. So now we will see the melting curve. Now you select the wells in the plate, which is here, and you will see that you have a primer of almost, amplicon of almost similar melting curve. So it means it is an example of a very good primer because it is not having any specific, non-specific binding or dimer. Whereas you go in this particular one and you will see still good, but there are some dimer. And how you can find that this is a dimer? Okay, this is perfect example. So in this particular example, you will see that there is a peak which itself is varying. It means that the size of amplicon may not be exactly similar, first thing. Second is that it is also having a small peak. So this could be because of the dimer. If the peak is not varying too much, then it's close to similar amplicon size. But if the peak is varying more than, close to 5 degree, so it could be an example of non-specific binding. So this is how you look into the software. Now that you are, and in this particular example, you will see that definitely there is a non-specific binding because you have one peak over here around 72 degree and one peak you have around 80 degree. So it means it's a non-specific binding. It means that the fluorescence intensity what you are getting for this particular reaction may not be coming from the target gene, right? So now that you have the file which can be opened in this Quant Studio Realtime PCR software, you can play around with it and you can click and then look and notice that which gene is having good specificity and which gene is having good efficiency. And tomorrow we can discuss about it if you have any doubt, right? So this is how it looks into the machine. Now, most of the analysis we prefer to do not using directly the machine software, but using either our own software or otherwise using the Excel sheet. For that case, you have an export option, which is the fourth option in the left side of the menu, and then you can click on the result. You will see that in the result, you have each well locations, sample name, target name, and then the city value. This city value, for example, you will see sample 1 GPX, okay, and then you have the city value, mean city value, standard deviation of city value, and then the quantity. So as I was saying that 1 microgram, if you, I mean 1 microliter, if you are using, then it is having city value of this particular thing, right? Everything can be exported directly into the Excel sheet, which you can do from exporting from here, right? See, something similar to this, which I have already shared, right? So now that was the whole story of using the Quant Studio software. We'll be doing again tomorrow for understanding how this is for the standard curve data. We will see how we can use it for the relative quantification. So for now, I am closing this particular window, and then you'll see that there is an Excel sheet named as standard curve, right? You open this. In the export result, you will see, so this is how it looks, right? I showed you now that how when you are downloading the data from the API, that Point Studio software, how it looks. So this is looking like this, right? To make some things clear and more understandable for you, what I did is that I copied the target gene name, this particular column, city value, okay, and the quantity in a separate sheet named as raw data standard curve. I hope you all can follow that. In here you will see that value is saved, right? So how do we actually able to find the standard curve using the Excel sheet, right? If you see, if you go into the third sheet where the calculation has been done, which I will be explaining it, which will make more sense to you. So if you see the raw data, you have three columns, right? You have gene name, DNA concentration, and city value, right? So you have gene name, DNA concentration, and the and then the city value. In between, you will see that there are two more columns. The first column is an assumption that 1 is considered as 100, right? It means if you are using the 1 microliter of the sample, then that corresponds to 100% of the sample you have put in that particular reaction, and that is in triplicate, right? Now the second one is you use, let's say, 1000 microliter, and then the second one you used 100 microliter. It means that you are reducing the 10%. So of 100%, what is the 10%? Is 10, right? Similarly, 0.02 we have used. So 0.02 of 1 is 2%, correct? Similarly, 0.01 of 1 is 1%. So this is basically, you have converted your cDNA concentration, which was there in the absolute number here, okay, in here, you converted into the percentage. Now, after you converted into the percentage, you can plot this particular and this particular city value. And let us see what it comes. It will come something like this, right? Which will not give you much information. Rather, if you convert the log, constant cDNA concentration from percentage to log value. So what is done in this? I will make it more visible. So now if you can see that column A is having the gene name, column B is having the cDNA, column C is converted the cDNA concentration into percentage, and column D is having log value of the percentage of cDNA, right? This one. And then the CT value you are getting directly from the machine, which is coming from the raw value over here, right? Now you select these two column, that is the log fold, log cDNA concentration and CT value, and construct a scatter plot, right? Once you construct a scatter plot, it will look something like this, where you have the log cDNA concentration into the x-axis and you have the CT value in the y-axis, and then you will be able to see that you get some points. Let us remove this line and we can put it again. So you go. So this is the linear regression curve. Line. So what you need to do is that you can put a red line. You click on the value which is plotted. You can click on the red line, and you can display the equation and the R square value. Now, in this particular example, what you will see is that it is having a slope value of minus 3.54. If you remember from the theoretical slides what I explained you, to be able to call it as 100% efficient primer, the difference between tenfold should be minus 3.3, right? So it shows minus 3.54, right? So even though it is having a good efficiency, it is not the 100% efficient primer. And how would you identify that? Is by calculating the efficiency using the slope value. So you have the slope value of minus 3.54 and you apply the formula, is that the 10 is to power minus 1 upon the slope value. So that tells you the efficiency. And the efficiency is always related to the specific primer. So this Excel sheet is already having all the formula in the columns. So if you click on the efficiency, you will see what it is. It is 10 to the power minus 1, minus 1 by slope value. And that gives you efficiency. So 1.91. If you remember well, then efficiency of 2 corresponds to two-fold change, right? Here it is 1.91. So how much is the efficiency? Is when you multiply this 1.91 by 100, that gives you almost close to 91% efficiency. So that is how you find out the efficiency. So basically you have to convert your cDNA concentration into percentage, convert into the log value, and then plot the log value and CT value, which will give you a linear regression curve. Now in this linear regression curve, you have the slope value, that is minus 3.54, which you write here, and this efficiency is 10 to the power minus 1 divided by this slope value, and that gives you efficiency. So this is how the efficiency is also calculated in ABI, that Point Studio software which you observed there, the graph which I showed in this particular case. So basically, if your efficiency is so this is how what I was saying, that this curve, no, let us go with just one GPX. So in this GPX, if you can see, it has calculated efficiency of 91.61, right? And in the Excel sheet, what we are getting of the same gene is 91.61, right? So it is exactly same. So now I hope that it is clear that how the efficiency was calculated in this particular graph. So this is how you calculate the efficiency in the Excel sheet. And then you simply multiply by 100 of the efficiency, that 1.91. It will give you 91.61 efficiency. So another way to understand, which is easy to understand, is that whatever it comes here, like 1.9161. So if you see after point now, whatever value, 91.61 is percent, I mean efficiency. Similarly, here you have 2.64, right? Since it is not 1 and it is 2, you have to consider that it is a highly efficient gene primer, which is not true, or which you cannot use it, but can be 64.37, and since it is 2, it is 164 percentage. Similarly, you have 2.03, so it is 103, right? So this is an easy way to understand that. And you always have to consider a primer of at least 90% efficient, and at the max 110% efficiency. So anything between 90 to 110, you can use it for your real-time experiment. But if there is anything which is varying too much, you should not use it, and better you should focus on designing a new primer. Okay, so this is how it is calculated. Now it is calculated for all four genes in the, I mean, one, two, three, yeah, three genes. If you want, you can remove the values and then you can try to calculate by yourself and try to make this graph for all the two genes which is present here. So similar, so what you need to do is, uh, so what you need to do is that, what I was saying. Yeah. So what you need to do is that you have to plot the same efficiency graph for the remaining these two genes, that is VVOP and tubulin 2. Okay. And all the formula is already written in the columns. So basically, this percentage has came that when you are you have the efficiency, you minus it with 1, which I said, and I mean which I said that after the point you have to consider. So basically, you have to minus 1 and then multiply it with 100. So whatever it comes, it gives you the efficiency. Right? So minus 1 and then multiply by 100. Correct? So easy way to understand is that if the efficiency, somebody is saying 1.9161, it should be around 91. If somebody is saying it is 1.6567, so it is around 65%. If somebody or if the efficiency is coming 1.85, then it is close to 85%. So similarly, you have to understand, if somebody is saying if the efficiency value is 2.31, then it is 131 percentage efficiency. So that those values you cannot take. So anything between 90 to 110 you can take. Right? Similarly, for the melt curve graph, in the export value, I don't know if I have copied here, but similar to that, if you check the export option of this particular table, you can see that there is a possibility to check to take the melt curve data. So melt curve data is you can again copy, and then there is one trick with this melt curve data is that there is a derivative value. It's a derivative value of the fluorescence. So basically, you measure the melt curve between 60 to 90 degrees Celsius based on some fixed values. As for example, here, if you find it's around 0.296 degrees Celsius between first and the second reading, and then you check how much the fluorescence is decreasing or increasing, and you define the derivative. There is some certain calculation by minus d and then fluorescence value divided by dt, and T stands for temperature over here. You get the derivative, and then you can plot your temperature value, and then you can plot your derivative value. So you will be able to see the melt curve. Since I have not selected everything, it's going like this. Oh, all the genes are there, so that's why it's 130 values for each gene. So if you can play around with this data, which is already shared with you, you will be able to see that how you can get the melt curve. So let us suppose you have this data with you. Oh, already it's plotted. Okay, so similarly, you have the for each gene, you can find out, you can separate the raw data like I was doing here. You can separate the raw data for temperature and the derivative curve. And so in the raw file, what you have is the value for the fluorescence and the derivative for fluorescence that is subtracted from the basal fluorescence values, as well as between the two temperatures, what is the changes in the fluorescence. Is having some formula which is using the derivative calculation, that is minus dt by, minus dx by dt. Uh, let's not go into the mathematical part of that. Straightforward, the concept is that you plot the temperature with the derivative melt curve values, and you will be able to see your curve. Right? So basically, you have the different. Okay. So as for example, the Excel sheet which is shared with you is having one gene C positioned at C1 well. It means that if you go here, which is your C1, your C1 is this one C1. It means that it's a GPX gene and replicate of 1000 template. I mean 100% template, and it gives you a peak curve here. So it means that if you are getting a single peak, it means that it is a targeted amplification, and it is not it is the fluorescence is not coming from the uh, primer, dimer, or non-specific binding. So I hope that it is clear by now.